Hello, good afternoon, my students. We thank God that we are coming back again today for today's lecture. Are you there? Good afternoon. Yes. Amen. Thank you, my brother. Thank you, my sister. Yes, thank you very much. Let us start. Thank you very much. In our last lecture, we did a summary or kind of revision of the lectures that we have done for the past three contacts. So we ended by looking at the prokaryotic and the eukaryotic cells. We gave examples of prokaryotic cells, the bacteria, and other unicellular organisms. We also gave examples of eukaryotic cells. These are the multicellular organisms. In the end, we finished them, and then I didn't give you assignment there. But one of the things we are going to do, which is a kind of assignment, is to, in a tabular form, I want you to give the differences, 10 differences between prokaryotic and eukaryotic cells. So that we'll summarize that chapter. So you have to give me 10 differences between eukaryotic and prokaryotic cells. Then we also look at the nutrition in bacteria. Bacteria is the eukaryotes, and you have different types of nutrition in eukaryotes or in prokaryotes. Example is the bacteria we are talking about. Bacteria is a prokaryote, so it have different categories of nutrition. The first one is saprotrophs. A bacteria is a feed saprophytically. That means they obtain their food from dead and decaying organic matter. So that's what saprotrophs do. They feed by obtaining their food material from dead and decaying organic matter. It's one of the feeding mechanisms in bacteria, which is a prokaryotic organism. Then another feeding method is by parasites or parasitism. This is where organisms live, either on the surface or inside the body of other organisms, derive nutrients from that organism and possibly cause harm to that organism. So it's one of the feeding mechanism in bacteria. Of course, bacteria are parasites. All we know that. Then another one, feeding mechanism, is also by mutualism. In some of the prokaryotes, they live in another organism, and then to the benefit of that organism they are living on, and also to their own benefit. So when two organisms live together and both organisms derive benefits from the association, then it is a kind of mutualism or symbiosis. So example is rhizobium that live in the root nodules of leguminous plants. They fix nitrogen in the root nodules of leguminous plants, enabling that plant to fix nitrogen and carry out the process of photosynthesis. In the end, they got protection from the root nodules. This is a symbiotic relationship. Another one is microorganisms that stay in the guts of termites. In the guts of termites, these microorganisms help termites to digest the cellulose that it takes, because termites feed on plants, and plants contain a lot of cellulose. So these bacteria or microorganisms that stay in the gut of termites help to digest the cellulose. On the other hand, they get protection from the gut of termites. So that gut of termites acts as the house or a kind of habitat to the microorganism. So this is a kind of symbiotic relationship. Another feeding method is autotrophic mechanism or bacteria. They carry out photosynthesis and use carbon dioxide as source of carbon. That's why we say photoautotrophic bacteria. So these are different types of feeding mechanisms. In prokaryotes, example is bacteria. So this is the summary, or that is the end of the prokaryotic and eukaryotic cells, the study of them. Today we are going to look at fungi. Fungi. The plural of fungi is fungus. Fungi belong to the group called eukaryotes, or eukaryotic animals or organisms, because they are multicellular. So we are going to look at fungi in this lecture. And at the end of this, our lecture today, as you have participated in this lecture, you will be able to draw and label a diagram of a typical fungus. You draw and label a diagram of a typical fungus at the end of this lecture today. Then you'll be able to list and discuss the characteristics of fungus. You'll be able to list and discuss the characteristics of fungus. And then we'll be able to differentiate types of fungi on the basis of their nutrition, reproduction, and biological importance. And finally, you'll be able to discuss the uses of fungi. These are the things we are going to do when we finish the lecture. So let us start. I said that fungi belong to the group called the eukaryotes. And then they don't have this chlorophyll. They lack chloroplast. And they are widely distributed in the environment. Examples of fungi are, you have Mucor mucedo, you have Penicillium notatum, then you have Rhizobium, then Rhizopus. Then you also have yeast. These are examples of fungi. And a particular field is dedicated to the study of fungi, and that is called mycology. So mycology is the study of fungi, and scientists that work on fungi or that work on mycology are called mycologists. So it's a very vast area of biology. So the study of fungi is known as what? Mycology. And scientists that are engaged in mycology are called mycologists. So we are giving examples of fungi. Let's now look at a typical diagram of a fungus. A typical diagram of a fungus shows that the fungus contains a kind of root-like structure that it's used to get itself attached on the host. And those root-like structures are called the hyphae. And when they become in bunch or connected, the whole root-like structure are called mycelium or mycelia. That is the root structure that they use to embed on the host. And then they now grow up. You now see a kind of something growing up called the sporangiophore. The sporangiophore contains at the tip the sporangium. And the sporangium contains the spores, of which when the fungi is matured, the sporangium can burst and release the spores. So I repeat, the roots that are embedded on the hosts are called the what? The hyphae. And when they become connected and embulged, it's called the mycelium. And then it grows up to form what we call the what? The sporangiophore, which at the top, it houses the what? The sporangium. The sporangium contains the what? The spores. And once the fungi get matured, something like this Mucor or Penicillium we are talking about, when the sporangium matures, it bursts and releases the spore. The spores can be carried by the wind, can be carried by contact. Anyway, then it disseminates the organisms and they now begin to multiply. Now let's now look at types of fungi based on nutrition. Types of fungi based on nutrition. So as I told you, you have penicillium as the example of fungi. You have mucor as the example of fungi. Remember that penicillium is a fungi that it can be synthesized to produce an antibiotic called penicillin. That penicillin you use as antibiotics comes from this fungi called penicillium. Remember mucor. When you leave your bread on eating for 3 days, maybe you forgot that you bought bread and kept it on the cupboard for 1 week. When you bring it out, you see this mucor growing on top of them. Even some of your shoes, when you leave them in a very moisty environment, you see this mucor, this fungi growing on them. So you also have the yeast. Yeast is a fungi. All these fungi we are talking about, they grow on stale food, rotting food materials, even soup that are spoiled, you see them growing on them. So the yeast also find unripe fruits. You find the yeast most used on ripe fruits. And because they are found on ripe fruits, they give the characteristic sauce, smell, or flavor in that fruit. So, and yeast are of economic importance because they can be used to manufacture what? They are used in brewery industry to manufacture different types of drinks. So these are the different types of fungi we have in the environment. So we have, as we mentioned, examples of fungi, the penicillium, example, the mucor, the rhizopus, yeast, etc. They are very plenty. These are fungi. Now, let's look at uses of fungi. What are the uses of fungi? Let's look at the different uses of fungi. One, yeast, for example, which is a fungi, is used in brewery industries for alcoholic fermentation. Is used in brewery industry for alcoholic fermentation. It quickens the fermentation in the brewing industry. So it's very, very important. And even in a bakery industry, it helps the dough to rise. Those of us that bake bread, those of us that bake different types of food materials, you find that when you add yeast onto it, the thing will rise. If you don't have yeast, I tell you that you will not make any breakthrough in the bakery industry because the thing cannot rise. So yeast helps the food material, the flour, to what? To rise. So apart from using yeast in bakery industry, it can also be used in brewery industry for fermentation. The second importance of fungi is the penicillium, which is of economic importance because it is used to produce an antibiotic called what? Penicillin. So penicillin antibiotics is synthesized from what? The fungi called penicillium. Now, another importance of, the third importance of fungi is that some species of fungi are used industrially to make cheese and organic acid. Some species of fungi are used to make cheese, bread cheese that you take. Now, another importance is that another species of fungi called aspergillus is used in the manufacture of alcohol from rice starch and in the manufacture of organic acids. So some fungi like aspergillus are used to convert rice starch into alcohol. This is very, very important because the rice starch can be used to make what? Drinks, to make alcohol. Then another importance of fungi is that some fungi like mushroom are delicacies. They can be used for food by man. So fungi like mushroom are important delicacies and used as food. In fact, if you go to a 3-star hotel and you demand for mushroom, you pay heavily for and before it is served to you. That's to show you that a lot of is of very great importance. And in several villages in Nigeria, people use mushroom in place. People that don't have access to meat, they harvest mushroom, and it's very, very delicious. I, in particular, have taken it. Very, very delicious. So, but you have to be very careful because some species of fungi are poisonous. So you don't just rush any mushroom to that it is eating. Some mushrooms are what? Poisonous. So you have the edible ones and the what? Poisonous what? Mushroom. So, but the most important thing is that some mushrooms are edible, and they are important delicacies as food, and use it in soup. So note also that fungi like chlorophyll, and so digest their food outside their body by absorbing the dead and decaying material into its body. This process of feeding is known as saprophytic mode of what? Nutrition. So all the fungi, they don't have chloroplast, and so they cannot manufacture their own food. So how do they food? They put the mycelium. The mycelium enter the host. They now digest the food material outside their own body and then absorb it. So when they digest that dead and decaying food material, they now absorb it and then grow. So that's how they do. Even the fungi that grow on some people's skin and hair, that is what they do. The place becomes very dirty, and so they now use the hypha or the mycelium on top of your body, and then do what? Digest the food material, dead and decaying food material on your body, and then absorb it. So that is it. That's why fungi can grow in some people's in our bodies. So having that said that, we are going to look at so that is on fungi. We are going to look at a close organism again called algae. So let us look at algae. Algae are plant-like photosynthetic organisms, and most are aquatic or live in water environment or moist environment. So they are photosynthetic because they contain chloroplast, unlike fungi that do not contain chloroplast. So they are classified, algae are classified on the basis of the pigments they possess. And so, of course, you now have what we call blue-green algae, the brown algae, the red algae, and then green algae. So these are how algae are classified based on the type of pigments they possess. So at the end of our lecture on this algae, you'll be able to draw a typical algae and label it fully. You'll be able to list and discuss the characteristics of two main groups of algae. You'll be able to differentiate between algae on the basis of nutrition, reproduction, and biological importance. And finally, at the end of our lecture on algae, you'll be able to differentiate between algae and fungi. This is very, very important. You'll be able to differentiate between algae and fungi. So let's look at a typical algae. And the typical algae we are going to draw is the chlorella. Chlorella is a typical algae we are going to draw. And the diagram is very simple. Just a kind of circular form of diagram. The first layer, the second layer. The first layer is called the cell wall. Just because cell wall shows that it is plant. And this cell wall is made up of cellulose. The second layer is called cell membrane. Of course, after the cell membrane, you have the cytoplasm. And inside the cytoplasm, you have a large area called the what? The chloroplast. The chloroplast, it dominates major part of the cytoplasm. And this chloroplast is the one that contains the chlorophyll that enables this chlorella to carry out photosynthesis. And of course, we have the nucleus. and the nucleolus in the corolla. Of course, we also have a vacuole, which is a very large. So this is the diagram of chlorella, which is the typical algae that we are discussing. Now, having said that, I want you to look at your manual, look at the chlorella. The outer is just a circular form. The outer covering is the cell wall. The second covering is the cell membrane. Then you now come to the cytoplasm. Inside the cytoplasm, you have a large area made up of chloroplast. Then you have the what? The nucleus. You have the nucleolus. And of course, I know that the nucleolus is inside the nucleus. And the nucleolus contains what? The chromosomes that contains the what? The gene. We all know that. Now, look, let's do a kind of practical. I want you to do this practical in your own leisure. And that practical is that you are going to go outside your compound and you are going to see ponds or a kind of ditches where water is collected. And then we look at it. Most of the ponds or ditches around your house, sometimes they turn green. That presence of green color shows you that algae is there. So you are going to collect the water, use containers you have to collect the water, and then you can now place them on slides and watch on the microscope. You will be able to see some of the algae we are talking about. You can see chlorella, you can see spirogyra. So you can see spirogyra. These are the major, some of the algae you find, especially when the rain is ending like this September, towards the end of September and the beginning of dry season. Most of the ponds, most of the water bodies you've seen start turning green, showing you that algae are plenty of algae there. And algae are very, very important, as we are going to look at the importance of algae later in our lecture. So I want you to do this practical. You collect them, then use your slides, do a kind of carry a pipette and put them on the slide and watch them under the microscope. Those of you that are opportune to have microscope in the house will be able to look at it. This is a practical. If you don't have them, when you go to study center, ask for the microscope. They will give you the microscope so that you'll be able to see some of these things we are talking practical. You will now see that the structure of algae is different from the structure of fungi because you see the chloroplast there, but in the fungi, you will not be able to see the chloroplast. So let us look at types of algae. So let's look at types of algae. Now, on the basis of their pigments, we can subdivide algae into the following groups. One is the blue-green algae. Blue-green algae. That's one type of algae, blue-green algae. The another type of algae is the euglena. Remember when we were in secondary school, we have talked of euglena. Euglena viridis is a very important algae we discussed in our junior secondary school. So euglena is a class, is one of the group of algae. Then you have what? The green algae. You have the green algae. That's another classification of algae or types of algae. Then you have the diatoms. You have the diatoms that microorganisms stay in water, but they have chloroplast. Then you also have the brown algae. The brown algae. Then you have red algae. So how many types have you mentioned? I've mentioned 6 types of algae. What is the first one? Blue-green algae. First one, blue-green algae. Second one, euglena. Second one, euglena. Third one, green algae. Third one, green algae. Fourth one, diatoms. Diatoms. Fourth one, diatoms. Fifth one, brown algae. Fifth one, brown algae. Then the sixth one is red algae. Sixth one is red algae. These are the different types of algae. So you have to note it. But note that all algae are autotrophic plants that manufacture their food on their own with the help of chlorophyll. Autotrophic. Autotrophic are those organisms that can manufacture their own food. We said that before. Those organisms that can manufacture their own food are called what? Autotrophs, because they contain chloroplast. Because they contain chloroplast, they can now manufacture food. Autotrophs are different trophs. Heterotrophs cannot manufacture their own food because they don't have what? Chlorophyll. Heterotroph cannot manufacture their own food because they don't contain chloroplast. So me and you are what? Heterotrophs. But plants are what? Autotrophs. That's what we are talking about. But remember, it's different from algae. Different from a fungi. Fungi do not manufacture their own foods, of course, but how do they obtain their food? They do their food by extracellular digestion. That means they digest the food outside their body and then absorb the food. But me and you, we digest our own food inside our body and then absorb it. So we are different from algae. We are different from what? Fungi. I hope you are following very well. Now, let us look at the biological importance of algae. Algae, are they of importance? Yes. Yes. Algae are very important. So we'll be talking of biological importance of algae. One, some algae serve as food. They serve as food materials. Yes. So some algae act as what? Food. Then two, algae serve a good purpose or a starting point in the food chain. It serves a good purpose or a good starting point in the what? Process of food chain. Why? Because some organisms feed on algae. So when they feed on the algae, another organism can feed on that organism. So they're now serving a very good, important purpose in the what? Food chain. Those of us that feed on animals or maybe fish. Some fish feed on algae, and then you can feed on that fish. So then, do you now see how it has served as a chain, a link? It has a good purpose in the process of food chain, because without that algae, fish will not be able to survive. Some fish will not be able to survive, but they survive because of algae, and you now feed on that fish in order to get the fish. So that is the importance of what? Algae in the water. So algae plays a very important role. Some have other various biological importance. Some can be used in medicinally to manufacture some important drugs. Yes, some can be used medicinally to manufacture some important drugs. So algae play a very important role in the process of food chain. Now, we can now look at differences between algae and fungi. Differences between algae and fungi. Can we remember some differences? You have pointed some out in the course of the lecture. Can we remember some of them? Differences between algae and fungi.
Yes, sir.
Yes. Yes. I want us to say some of the important differences.
Sir, algae has chlorophyll and fungi does not have chlorophyll.
Very good. Thank you very much. Thank you. Ibrahim, Fatima. Yes.
Yes, sir.
Yes. Is it Ibrahim that answered this question? Okay, Fatima Osman. Okay. I want more contribution. Yeah, James Madu Abuchi, you want to say something?
I will help you first. Yes, sir.
James Madu Abuchi, yes, you want to say something?
Yes. I'll just have to.
Hello, okay, sir.
Yes, I'm listening.
Algae cell wall are true. They have
Yes.
cell wall
This,
through cellulose, while
yes, cellulose cell
fungi
wall.
have
Very
cell
good.
wall chitin.
Very good. Thank you very much. You have been able to say that. Yes. Yes.
Hello,
Yes,
sir.
I'm listening. Yeah.
Algae reserve carbohydrates, fungi glycogen.
Okay, okay. Fungi reserve their food in form of glycogen. Yes, Ibrahim Fatima. Yes, say something.
Algae are autotrophic
Ibrahim.
and fungi are autotrophic. Sir.
Okay, very good, very good.
Algae are autotrophic. Yes, sir.
Yes, yes, yes. Fatima Moletala, say something.
Yes, sir.
Yes.
Algae lives in water, while fungi live in as parasites.
Yes, yes. I want somebody to say something in terms of method of feeding, making food.
So I want to say something.
Yes. Ayog can say something.
Algae are autotroph. They make food by themselves, while
Yes.
fungi are heterotroph. They depend on
Yes.
other things to feed.
Thank you very much. You are very, very good. Christopher, say something. Christopher, say something. Christopher. Rose. Okay. Can we continue? Christopher,
Yes,
say something. Yes, yes. Okay. Yes, yes, yes. Emmanuel de Santos, say something. Okay. Let's look at the differences.
Yes.
So let's look at the differences. So one of the first one is that algae contains the chlorophyll, as one of you suggested, but fungi do not contain chlorophyll. Then algae is autotrophic. That means they manufacture their own food, just like any other plant. If you look at that dogoyaro in your compound or that mango in your compound, they are autotrophic because they can manufacture their own food because they contain the chloroplast. And so during the process of photosynthesis, what happens? You see, they utilize carbon dioxide, then they utilize water in the presence of sunlight and chlorophyll. Then they now manufacture what? Glucose. So and then give out oxygen. So they are autotrophic. Algae are autotrophic because they can manufacture food. And as they manufacture their own food, they release oxygen to the atmosphere. And that oxygen they are releasing are taken by what? Animals or heterotrophs in order to survive. So algae are autotrophics, while fungi are heterotrophs because they feed on the food manufactured by other organisms. So you are autotroph because if you are eating beans today, if you are eating maize or corn today, then or even bread today, you are not heterotroph because you are eating the food manufactured by the autotrophs. So that's that. Then the third difference is that algae have a true parenchymatous tissue. Then fungi have false parenchymatous tissue. They have hyphae, or what we call the mycelium, which is not a true root. Now, algae have what? Cell wall, true cell wall. They have cellulose cell wall that gives it the rigidity, that give the cell the rigidity. But fungi have what? Cell wall made up of chitin. Then algae live in aquatic environment, water or wet substrates. They live in aquatic environment. But fungi live as parasites, either as an ectoparasite or endoparasite, or a kind of hyperparasites. And hyperparasites are those parasites that lead to the death of their host. It's different from normal parasites because normal parasites do not allow the host to die. Because if the host die, they are already gone too. So they tend to maintain a balance whereby they continue to feed on the host. And that's why that tapeworm in the body of a human being will continue to live and survive and gradually take nutrients from you so that they will continue to live in your body for a very long period of time. So the ordinary parasite. But a hyperparasite is a parasite that leads to the death of their what? The host. So when a parasite now leads to the death of the host, that is a hyperparasite. So while algae stay in aquatic environment, then fungi live either on surface or inside another organism. Now, algae reserve carbohydrates in form of starch because the end product of digestion of when they manufacture their food, they reserve it in form of what? Starch. And that's why maize store starch, cocoyam store starch. Most of these food materials, they store their food in form of what? Starch. So they reserve their carbohydrate in form of starch. But fungi reserve their own in form of glycogen. Reproduction in algae is by cell division or asexual or sexual reproduction. In fact, I don't know, in your secondary school, you have talked or they taught you about the reproduction of algae, something like a spirogyra. You see how it divides and cuts, either by cell division, either by asexual or sexual. So we did that in secondary school. Even in conjugation, we all did that in secondary school. Now, for fungi, they reproduce also vegetatively by asexual spores. They liberate the spores. The sporangium contains the spores. And once the sporangium matures and bursts open, it liberates the spores. And then that is how the spores are carried away. And it can be carried either by the wind, it can be carried by body contact, it can be carried by not obeying personal hygiene. When somebody who is having a fungal attack or has fungi in the body, and then when you use the beddings or use the towels or use the inner wears of somebody that has this type of fungal attack, definitely it will come to you. And that's why in most school children, you see a kind of fungal attack. Many of them go down with fungal attack because of their mode of what? Playing and the mode of interaction. They will contact it, especially the one that attacked the head. Before you know it, all the children in that class will have the fungal attack because the sporangium bursts and liberate the spores. And in the course of playing and interaction, the spores will do what? They will contact the spores and carry it, and then it gets spread. So if you use clothing of somebody who has fungi, then definitely it also come to you, especially the females. Remember that these fungi are found in the private parts of individuals. So if you use same material or same clothing, definitely you go down with the fungal attack. Let us use the next 5 minutes to do a summary of what we studied today. The attendance was very poor, only 58 attendance, unlike the other time when we have about 200 attendance. Is it because I did not communicate you?
Yes, sir.
Yes,
A
sir,
lot
many
of
were not
people.
aware.
A lot of people are not aware.
But we are aware that our lecture is Thursday.
Yes,
Yeah,
sir.
but it doesn't show on our portal anymore.
Okay, sorry. From this week, I will continue to send information to you. The lecture is 3 o'clock every Thursday.
Like I just
Is
have
that
to
okay?
copy the links now and invite many people now to join. Yeah, thank you, sir.
Okay, thank you very much on what we have done today. We started by looking at the prokaryotes, which we summarized last week, but we carried over assignments, which we did major differences between prokaryotic and eukaryotic cells. So that you should do that. 10 differences between prokaryotes and eukaryotes. And today we also refreshed our mind back on the nutrition in bacteria, certain categories of nutrition in bacteria, which is the prokaryotes. We talk of saprophytic nutrition. Just as you have in fungi now, we are now having saprophytic nutrition in bacteria. Yes, because they can live on dead and decaying material and then absorb it. Then you also have parasitism as a mode of nutrition in bacteria, because parasite is where an organism lives in another organism. They have nutrients in that organism and harm that organism. Some bacteria are a kind of symbiotic relationship. They are in a kind of symbiotic relationship, or what we call mutualism. Here, the two organisms live together for the mutual benefit of one another. Both organisms benefit from the association, just like as you have in the rhizobium, which is a microorganism in the root nodules of leguminous plants. They help to fix nitrogen in the root nodules for the organism to, for the leguminous plant to produce fruit, roots or food. Then, so the rhizobium, that bacteria, helps that organism, help the nitrogenous plant to fix nitrogen. Something like granite. Granite is a leguminous plant. So in the root nodules of this leguminous plant, you have what? Rhizobium that helps to fix nitrogen so that these legumes will be able to do well. So in the end, they give protection to the bacteria, to the microorganism or the bacteria. This is an example of mutualism or symbiotic relationship. Then we also talk of photoautotrophic bacteria that use carbon dioxide as a source of what? Carbon, to give it energy. From there, we went to fungi. We look at fungi. We said that fungus or a fungi belong to the group called eukaryotes. Organisms, because they are multicellular, but they don't have chloroplast. They don't have chloroplast. However, how do they feed? They feed on dead and decaying material and then digest the dead and decaying material outside its own body and then absorb it. They use the mycelia to digest that food material and absorb the nutrients into its own body. So we also say that we gave example of fungi like mucormycoides, penicillium natatum. You have rhizobium nigrica. Then you also have yeast. These are examples of fungi. Then we drew a typical diagram of a fungi or a fungus, and we say that the typical diagram shows that it has roots, but we don't call it. We will call them hyphae. And when you have a bunch of hyphae, they make up a mycelium. Then they have a stem called what? The sporangiophore. And the sporangiophore has a kind of top in a kind of that look like a
Vertically growing hyphae.
cup that contains the mucor, the rhizobium, the yeast, and they grow on stale food, stale bread, rotten food materials, and then rotten fruits. That's where you find mostly the yeast. Now, then to manufacture alcoholic food by what we call alcoholic fermentation. Some of them, like penicillium, are used to produce drugs. Like as you have penicillium is used to produce an antibiotic called penicillin. Then some species of So some species of penicillium are used to make cheese. They are used in making cheese in bakery industries. Some species of aspergillus are used to manufacture alcohol from rice carbohydrates. So they can also be used in manufacture of alcohol from rice carbohydrates, and also used to manufacture organic acids. Some species of fungi like mushroom are edible. They are edible. It can be eaten. And then there are delicacies in some places, especially in some hotels. So, and we summarize that fungi, generally, all of them, they lack chloroplast. And because they lack chloroplast, they cannot manufacture their own food. What they do is that they digest dead and decaying material outside its own body using this mycelium or hypha, and then absorbed into its body. This process of feeding is called saprophytism. So saprophytism is the process of feeding whereby the organism digests dead and decaying material and absorb it into its own body. Then we now look at algae. We say that algae are plant-like photosynthetic organisms, and most of them are aquatic. And then we drew a structure, a typical structure of an algae, and then we use chlorella as the typical structure or diagram of algae. And then in that diagram, the outermost covering is the cell wall, cellulose cell wall, which we know. And then the cellulose cell wall is followed by the cell membrane, and then followed by the cytoplasm. And inside the cytoplasm, you have a very large chloroplast. And then you now have the nucleus. And then you have the nucleolus inside the nucleus, and preferably you have the vacuum. This is the diagram of chlorella. Then we also encourage you to do a little practical. I encourage you to do practical, whereby you go outside your building in that pond or ditch that are almost turning blue. You use a container and collect the water, and then put that water, use a pipette and put on a slide and view through the microscope. What you will see will surprise you, because you see different fungi, different algae in that collection. And so when the pond or ditch is turning blue or greenish, it shows you that algae are there. And I'm very, very convinced that you can find it even in your environment there. So you need to do that to be able to see even the spirogyra we are talking about. You see it clearly shown, and they are in chains. And other algae you can also observe. Even chlorella, you can see that. So we now classify, looked at different types of algae based on the pigment they have. So you have 5. Okay, we listed 6 types based on the pigment they have. You have the blue-green algae, you have the euglena, you have the green algae, you have the diatoms, you have the brown algae, and the red algae. And we say that all algae are autotrophic because they manufacture their own food with the use of chlorophyll. And then in the process of this photosynthesis, oxygen is released. So the algae also help to purify the environment. Yes, it has to purify the environment. Yes, under climatic change that is happening now, algae can be of very good importance because they help to purify the environment. They have to reduce the amount of carbon dioxide in the atmosphere. And those of us that are into aquaculture, you can use algae in aquaculture. So these are some of the importance of algae. And then I also say that the algae have a very good link in the process of food chain, because some organisms eat algae in the food chain. And remember that food chain is the process of eating and being eaten, where one organism eats another organism and another organism eats that one. So the process of eating and being eaten is food chain. And a complex form of food chain forms food web. So algae plays this very important role in the food chain. Now, finally, we looked at the differences between algae and fungi. Under algae has a chlorophyll, but fungi do not have chlorophyll. Under algae, Algae are autotrophic, but fungi are heterotrophic. Algae have a true parenchyma, then fungi have false parenchyma, that's hyphae. Algae have cell wall, which is true cellulose. Fungi have cell wall, but they are cheating. That's the chitinous cell wall. Algae live in aquatic or water environment or wet environment. Then fungi either on or under the body of other organisms. Then algae reserve their food as carbohydrates. Fungi reserve their food as glycogen. Reproduction in algae is by vegetative cell division or asexual division or sexual division. But that of fungi is by vegetative or sexual spores, formation of spores. That is where we have it today. I want you to ask questions. Any questions for me? So I want, yes.
There's question.
Yes.
Sir, you talk about the diagram, the structure of algae, but I didn't see it on the screen. Like you said, you will draw it.
Okay, then see it in your manual. You have your
Okay.
manual, you see it there. Any other question?
No, that's for now.
Okay.
Sir, I want to ask. So the topic of today now is based on algae, right?
Algae, fungi, and we completed a lecture on eukaryotic cells, and then we tell you to do the 10 differences between prokaryotes and eukaryotes. Mwangele, ask your question. Mwangele, ask your question. Or Ayog.
Good afternoon, sir.
Afternoon, how are you?
Fine, sir.
Yes.
Sir, please, I don't used to know the difference between blue-green algae and green algae.
Okay. Yes, if you go into serious hydrobiology, you now see that there is a difference between blue-green algae and what? And ordinary algae. Like this chlorella we are talking about is ordinary algae. The Spirogyra is a different algae, is ordinary algae. Then you also have blue-green algae, which you can find in a aquatic habitat. So those of us that are in hydrobiology and fisheries or aquaculture can tell you better. But for you now, some of you are in computer. We don't want to go deep. Is that okay? Matthew, ask your question. Matthew, ask your question.
That's the same question I wanted to ask. She asked already.
Thank you very much. Thank you very much. Okay. Any other question before we round up? We have just 1 minute to play.
So do we have assignments, sir?
Yes, now 10 differences between prokaryotic and eukaryotic cells. Don't go and copy the manual. Do it on your own, because I see that most of the assignments are given,
sir.
some of you went and copied the manual. So
Sir,
next
please,
week,
aquaculture. I'm doing animal science, so I want to know the main difference, sir.
Okay, you are in aquaculture.
Yes, as in I'm doing animal science.
Okay, that would be a very good area for your study, and we are going to meet more in aquaculture studies. I teach you aquaculture. Next week, Thursday, don't wait for me to call you. The time is 3 o'clock. You join us. And when we come, we are going to look at roots. So go and look at roots, the root system. That's what I'm going to do on next week, Thursday. So don't wait for me to give you a link. The link is the same. Yes?
Okay, sir.
Hello, sir. Please, so this your class was not in our portal today, so I joined late, sir.
Okay, next week I will send the link to you. Oh, thank you,
Yes,
and God bless you.
That's why the assignment, sir. Regarding the assignment, are we joining anything before we list the 10 difference?
No, just give me the 10 differences between.
That's all.
Yes, yes. Don't go and copy in your manual.
No.
Go it and do a lot of research. Thank you. That's why we are going to end it today. And next time we are going to meet next week, Thursday, 3 o'clock. Goodbye.
Okay, bye-bye, sir.
Bye, sir. Thank you, sir. I appreciate it. Yeah, bye-bye.
Bye.
Thank you.
You, sir.
What is assignment, please?